So building a model, an atomic model into electron density map is probably the most exciting step of this whole experiment, because you get the feeling that you are the first one to reveal a molecular structure that no one else seen before, ever.
Alexey Amunts is head of the cryo-EM laboratory at SciLifeLab and a scientist at the Institution for Biochemistry and Biophysics at Stockholm's University. The main focus of his team is on trying to find the answer to how proteins are synthesized in organelles in the human cells and visualize them with an electron density map.
I did a PhD in Tel Aviv University on the structure of plant photosystem 1. And then moved to Cambridge MRC Laboratory of Molecular Biology to work on the structural biology of ribosomes. Now, during the postdoc period, the electron cryomicroscopy developed to the level where it can solve high-resolution structures. And the next stage for me was to establish my own lab, where in a way I combined these two interests of photosynthetic complexes, large bioenergetic macromolecules, together with the ribosomes, the protein synthesis machinery. And now we investigate how proteins are synthesized in organelles. So we tackle very fundamental processes, and particularly this process of protein synthesis in mitochondria is central to all advanced forms of life. Without proteins in mitochondria, we would not have the world as we know it. The interest of my lab is the molecular mechanisms underlying cellular bioenergetics. How the genetic information is read and how it is translated into functional bioenergetic units, that is the question that we're trying to investigate in the lab. Now, in the lab, we work with different types of cells. Human cells, human cancer cells, different parasites, and also plant photosynthetic cells. Before being able to visualize a molecule, it first should be isolated from a cell and purified from other types of molecules. The first step, we break the cells, and once they are purified to a high degree of homogeneity, then they can be frozen in a dedicated cryo-EM lab and subjected to electron microscope. I think any process that you want to understand, there is no better way than directly visualize it. And cryo-EM at the moment is the most dominant technique and the highest resolution technique that allows visualization of molecules. So now we look at the frozen grid in the electron microscope. Now you can see that the grid is composed of multiple squares. And if we zoom in into a single square, we can see that it's composed of multiple holes. If we zoom in further into a single hole, this is where our molecules are trapped in a thin ice sheet. And those are the molecules that were previously purified from cells. Now, these images are quite noisy. This is not a high-resolution image. To increase the resolution, we collect thousands and tens of thousands of such images and then computationally align them onto each other and extract the particles, which then leads to generation of the three-dimensional model of the molecule. I think what is most exciting for me at the moment is that we're trying to answer this big enigma in the field of how proteins are synthesized in organelles, and we combine it with the most cutting-edge technology. So this combination of answering a new question and using very high-tech technology is attractive.